Good morning or good afternoon, everyone, depending on where you are joining us from, and welcome to today's webinar. My name is Rachel Starbuck from the Business Review, and I will be your host. It is our pleasure to have Piramal Healthcare with us today, who will be discussing Doing Difficult Chemistry by Harnessing Biology. Today's guest speaker is Bob Holt, who is a Biocatalyst Director at Piramal Healthcare. I'd like to welcome you to our new webinar platform. You'll notice that this webinar is browser-based, so if you disconnect, please click on the link that you received via email to rejoin. You may move and resize the windows on your screen to customize your view. All of the icons along the bottom of your screen are the interactive widgets that we have on offer today, so please do interact with them all throughout the session. In order to ask questions, you can send them in via the Q&A widget at the bottom of your screen, or please use the top left-hand box. We will allocate around 10 to 15 minutes at the end of the session to address any questions or thoughts that you may have. Please use the help widget if you experience any technical difficulties or require any more assistance. So without further ado, please allow me to welcome Bob.
Thanks for the introduction, Rachel, and thanks to everyone for attending. I hope you'll find the presentation interesting. I hope that the title is self-explanatory. What I'm going to try and introduce to you today is how we can use biological systems to do some clever chemistry. Biocatalysis is a powerful technology that's been developing rapidly over the last 25 years. Initially, it was viewed with skepticism by the process chemistry community, but now seems to be firmly embedded in the synthetic repertoire of Piramal Pharma solutions. During the presentation, I want to illustrate how we were able to demonstrate the value of biocatalysis with some real industrial examples. But before launching into the technical part of the presentation, I want to give a brief overview of Piramal Pharma Solutions as a company. So Piramal Pharma Solutions is a division of Piramal Healthcare. We're a global contract development and manufacturing company, and providing innovative services and for the generic pharmaceutical companies worldwide. We provide services across the whole of the drug development process, starting with discovery, where we are involved in the development of lead compounds, intermediates and API development and manufacture, drug formulation. We have specialized technologies such as antibody drug conjugation as well, which is operated out of one of our UK sites. And we also have sterile fill finish and formulation facilities. So we're covering the whole drug life cycle from the initial invention of a new chemical entity through chemical development, through formulation, and finally through to packaging and so on. So in support of that, we have a global manufacturing network. Assets spread all across the world, from the US, in Canada and the US, through Europe, and also into India. And this allows the strategic placement of projects to take advantage of lower costs, local supply chains, or specific customer or market requirements. As an innovative company, R&D is very important to us, and we're looking to provide the optimum solution to our clients. In order to do that, R&D plays a very important role. Our vision for R&D is to be the best R&D group servicing the CRAMS and API generics businesses headquartered in India. Our aim is to develop safe and green manufacturing routes, providing a cost advantage. If we don't provide a cost advantage, then we're not going to be successful. So that's always a prime objective. What we want to do is to create a culture that promotes creative thinking by all of our scientists, not just group leaders and directors. We're trying to get everybody engaged in the final objective of coming up with clever ways of solving chemical synthesis problems. And what we're hoping is that will lead us to be the company of choice for scientists with high ambitions for career progression in an R&D environment. We're aiming to develop a defined portfolio of technologies to support our core chemical capability overlaid on that. So what's our technical philosophy? Obviously, our focus is on scalable technologies. Everything we do is about manufacturing. We don't want to be distracted by clever chemistry which is not commercially viable. So scalability is something which is in our minds right from the very first inklings of a project. We've always got our mind on the end game. How do we manufacture this at a ton scale? Speed is obviously important, as is cost, almost goes without saying. One thing that we have is an unbiased approach to the application of technology. What we're after is the most efficient route of synthesis, and it doesn't matter where we get the technology from. It's nice when we can develop it ourselves, invent it in-house, but we're not constrained by a not invented here syndrome. If somebody else has a better technology or there's a free-to-use technology, then we will want to take that, because ultimately we want to come up with the most cost-efficient and most efficient route. In terms of using other people's technology, intellectual property is always an issue, and we would never ever infringe on intellectual property. We take a very strong stance on that as a company, and of course we will license technology from other people who hold IP where that's appropriate. Where we can, we will use commercially available catalysts. No point in reinventing the wheel. Speed is important to us, so we want to move as quickly as possible. However, where we need a novel catalyst to develop a new technology, then we will develop novel catalysts ourselves. So, moving on to our technical capability, we have an underpinning comprehensive chemistry capability. That's the core of our activity. What we're about is doing chemistry. We're moving from discovery scale to hundreds of tons. And to kind of support that, we have a global pool of about 350 R&D scientists, 75 of which are at PhD level, all within the synthetic group. Quality compliance goes without saying. We have to apply the appropriate level of quality to whatever we do, and whatever stage of the life cycle of that compound, whether it's in discovery or whether it's in manufacture. Overlaid on top of this core capability, we have a number of specialized techniques. These include asymmetric chemical catalysis, continuous flow chemistry, and also biocatalysis. And the key thing to all these technologies is a seamless integration of the specialist technologies into the overall synthesis. Everything has to go together to bring us through successfully to the end point. So, for the remainder of the presentation, I'm going to focus on biocatalysis itself. And I want to start off by asking a question. What is biocatalysis? Different people will have different opinions of this, different answers to the question. And it can be answered in different ways. So the way I'm going to define it today is simply, it's the application of biological catalysts in organic synthesis. And when I say biological catalysts, really what I'm talking about is enzymes. Now, the enzyme may be used as a single enzyme in a purified form, perhaps. It may be a reaction catalyzed by an enzyme within a whole cell. And it may be a single-step reaction, and typically that's what we are talking about. We're converting compound A to compound B using an enzyme as catalyst. But it could also be a more complex sequence of reactions catalyzed by a series of enzymes. For the purposes of today's presentation, we're going to be talking about single-step reactions. So, next question that arises is, why use biocatalysis? Well, what you can see here in the bottom right corner of the screen is a representation of a cytochrome P450 enzyme. So this is an enzyme. The colored ribbons that you can see in green and blue represent the protein backbone, and the brown structure in the middle is a heme group, which is a porphyrin structure with an iron atom ligated at the center of it. Molecular weight of this thing in total is about 110,000 Daltons, so it's quite a complex and quite a nice-looking catalyst when you see it in this format. But organic chemists have been catalyzing reactions with things as simple as a proton or iron filings or something like this for decades. So why on earth would you want to use something as complex as this enzyme? Well, you can answer that question with one word, and that word is selectivity. Selectivity itself can be broken down into a number of subsections, and the first one is stereoselectivity, and this is probably the major area where enzymes have come into their own. But there are other areas of selectivity where enzymes can be very useful. The first is regioselectivity, and another one is chemoselectivity. And throughout the talk, I'm going to give examples which highlight these particular aspects of enzyme catalysis. Other advantages that enzymes bring are that they operate under mild reaction conditions, typically ambient temperatures, by which I mean in the range 20 to 40 degrees Celsius. Around about neutral pH generally. Typical range for the enzyme reactions that we would operate would fall in the window of pH 4 through to about pH 9, maybe 9.5, but really not outside those ranges. Water is often used as a solvent, and that can be advantageous in avoiding the use of organic solvents, which can be considered as potential pollutants and need recycling or disposal. Having said that, many enzymes can tolerate organic solvents, and the use of enzymes in organic solvents does occur, and I will refer to that in some of the examples later. It avoids the use of toxic reagents. Very often, chemical reagents will contain heavy metals, and by using a protein as a catalyst instead, you avoid that. A very wide range of reactions can be catalyzed, and this is really important. Biocatalysis is not a narrow capability. There's not many things that you can't do with enzymes if you really think about how to go about it. When you consider that some microorganisms can build up all of their cellular structures, their nucleic acids, their proteins, their vitamins, their coenzymes, all of these things can be built up from some inorganic salts and a simple carbon source like lactate or glucose. In order to use that simple carbon source and build up these complex structures, must entail the use of a wide repertoire of chemical capability. And they're the enzymes that we're trying to exploit in biocatalysis. Finally, scale-up is generally straightforward. You don't have to worry about exotherms. If you can do a reaction in the lab at 100-gram scale, a liter scale, generally you can scale up to thousands of liters straight away. No need to go through intermediate steps. You may want to do it just for comfort's sake, but generally speaking, there's no real need. So I came into the industry in about 1990, 25 years ago. What was the state of biocatalysis back then? Well, to be honest, most chemists considered biocatalysis to be an academic curiosity. Very few enzymes were available at commercial scale, and most of the ones that were related to the food and detergent processing industries, so proteases and lipases primarily. Very few chemists would consider biocatalysis, and when they did, it was only when all other options had failed. It was like a last resort. Most process chemists viewed biocatalysis as black magic. So what's happened to change all this? I'll go through a couple of examples that I've been involved in, and these are compounds, problems which we solved, which helped to change the perception amongst our own company. The first one is the synthesis of a triazole antifungal compound shown here. Originally synthesized as a racemate, remember this was back in around 1990, around about the time that the understanding of the importance of chirality to biological systems was just coming through, new regulations coming into the pharmaceutical industry, dictating that if a compound was racemic, it would only be acceptable to make it as a single enantiomer. So this drug was initially conceived of as a racemate. The synthesis was developed, and then it was necessary to make a switch to the synthesis to now make the enantiomer. So various chemical options were considered, various things were tried, but without success. Things could be done at milligram or gram scale, but nothing that was scalable. There was a desire to stay as close as possible to the original synthetic route because of all the development that had gone into this relatively complex molecule. The key intermediate, the first chiral molecule in the synthesis, was the triazole epoxide shown here. So the idea was to try and find a way to make this in its optically active form. The chemists tried sharpless asymmetric epoxidation approach, but unfortunately it simply wasn't productive enough or scalable. So this is a point at which collaboration with the biocatalysis team was established. It was typical of the situation I mentioned a moment ago. When all else fails, let's have a look at biocatalysis. So we took a look at this, and initially we considered could we find a biological replacement or alternative for the sharpless epoxidation and do a direct epoxidation on the alkene. Looks nice on paper, but unfortunately at that time there were no catalysts available commercially that would do this job. We identified whole microorganisms that would do the reaction, but the productivity was very, very poor. We also considered a resolution of the epoxide using an epoxide hydrolase. And again, although we could demonstrate the concept, the reactivity, the reaction rates and productivities were very poor. So we had to look for an alternative way of dealing with this problem. But what we did was to take a slight deviation, and starting from the racemic epoxide, we did a chemical hydrolysis to open that to the dial. Once we got the dial, we could selectively esterify the primary alcohol because the tertiary center is pretty unreactive. So we've got a nice, clean, selective esterification at the primary center. So this now gave us a substrate which was amenable to resolution by a lipase enzyme. At the time that we did this work, there was only about 12 enzymes available at commercial scale. And through a screening process, we identified an enzyme, in fact it was pig pancreatic lipase, which would catalyze this reaction shown here to give the desired enantiomer as the residual substrate, and the yield and conversions were around about 42% isolated yield, 98% EE. So a pretty good resolution. We could then take this residual ester and convert it back to the epoxide and plug that back into the existing synthesis. So the way this reaction was done was by dissolving the racemic ester in toluene, dissolving the enzyme in aqueous phosphate buffer, and mixing the two together as an aqueous organic two-phase system. The advantage of this was it allowed us to achieve high reactor concentrations for the substrate because the ester was pretty much insoluble in water. And the converse, the product of the reaction, the diol, was pretty soluble in water. So at the end of the reaction, we simply stop agitation, allow a phase separation, decant the aqueous phase to remove the diol, and effectively recover almost free of residual diol, the ester here. A simple water wash of the organic phase and recover the optically pure ester. So very quickly we were able to scale this up to 100 kilograms and to use that material in support of clinical trials. And this success really started to change the opinion of some of the organic chemists who initially were skeptical. We'd managed to help them with a problem they couldn't solve themselves, and suddenly we started to get a flood of inquiries. So that was our first real success in trying to turn the tide in favor of biocatalysis. One interesting aspect that is worth pointing out here is that the actual center which is reacting is here, the carbonyl on the ester group. It's actually distal from the chiral center. So the actual enzyme reaction doesn't have to occur at the chiral center of interest. It can be remote, and the enzyme may still recognize that. So the next example I want to talk about is the synthesis of a 2-methyl tetrahydropyranone. I've actually abbreviated the name just for simplicity on the slide here, so please forgive me for that. So the final target we're aiming for is this 2-methyl tetrahydropyranone. Not many handles on this for doing any enzymatic chemistry, but fortuitously this molecule is accessed from the corresponding alcohols. And when you make these alcohols, although there are 2 chiral centers, so theoretically 4 potential diastereoisomers, because of the diastereoinduction, you actually end up with a mixture of enantiomers rather than diastereoisomers. So the problem reduces to a separation of enantiomers from this mixture. The hydroxyl handle, of course, here offers us another opportunity to use an ester hydrolase enzyme to catalyze the resolution. So learning from what we just described in the previous example, we can take a similar approach. So starting from the racemic mixture of pyranol shown here, we can do an esterification reaction, form the racemic mixture of the acetyl or butyryl ester. And we then screen these with a range of commercially available lipases to look for a selective reaction. And in this case, we were able to identify the enzyme Candida Antarctica lipase B, also known as Novozyme SP435, very active enzyme in an immobilized form. Did this reaction with great selectivity to very low catalyst loading. We were able to recover the desired enantiomer as the unreacted ester in 45% yield, 98% EE, and then we could deasterify and oxidize to give us our target compound. So again, we've taken a bit of a circuitous route to get from where we started from to where we want to be. But the question that springs to mind is, can we cut out some of these steps? Now, when you look at what's happening here, the enzyme is actually recognizing this configuration and it's hydrolyzing it. So we reason that if that's the case, if we could run the reaction in the reverse direction, i.e. in a transesterification mode, we would expect this enantiomer to react, and therefore for the undesired enantiomer to be converted to the ester, and the desired enantiomer to remain unreacted. And in fact, this is what we see. So by doing the reaction in the transesterification mode, we can cut out this initial esterification step and the final deesterification step and remove 2 steps from our overall synthetic pathway. And this reaction works extremely well. In fact, we don't even use a solvent. So we do this reaction simply by mixing the starting material Piramal, which is an oil, with the vinyl acetate or vinyl butyrate, which is also an oil, sprinkle in the solid enzyme, which is immobilized, and stir it up for 4 or 5 hours. The reaction runs to completion, and then you simply extract the Piramal into water. So we're actually running a solvent-free transesterification operating at 60% weight for substrate. So this helped to dispel one of the other myths about biocatalysis, which was chemists believed that reactions had to be done in water, which they don't, and B, they were always dilute, which they're not. So again, this is a good example of demystifying things for the organic chemists and helping to get them on side in terms of using biocatalysts. So, both of the reactions I've talked about so far, very clean, very selective resolutions. So in that sense, we're very happy with ourselves. We've done a better job than we could do with conventional chemical resolution. But at the end of the day, there's still resolutions. We're still losing over 50% of our starting material. Can we do anything to improve on that situation? And the answer is yes. We can actually apply what we call Desymmetrisation technology. So cast your mind back to the first example with the triazole. We can actually generate a symmetrical precursor to that triazole, which is shown here. So we have a diester formed here, which is symmetrical, no chiral center, but it is prochiral. So if we do a reaction at one or other of the prochiral centers, the molecule becomes chiral. What we were able to show is that by using a particular lipase, we could hydrolyze selectively one of the enantiotopic esters to give us an enantiomerically enriched monoester. In this case, about 90% yield of 96% enantiomeric excess. Unfortunately, the enantiomeric excess isn't as high as we've seen in the previous reactions and probably isn't acceptable in terms of the kind of optical purity required for an API. But we can convert it through to the epoxide, and if our end product is a compound which contains additional chiral centers, such as this example here, posaconazole, which is a current target of ours, it does give us the option to purify by crystallization of diastereoisomers at a downstream step. What I'm showing here on the right is that you also get some overreaction here, so you get hydrolysis of the second ester group to yield triol, which effectively diminishes your yield from the 100% theoretical maximum. But of course, if you want to, if there's a significant amount of overhydrolysis, this can be plugged back into the synthesis and converted back to diester and recycled. So desymmetrisation is a very, very attractive approach. And if you can build that into your synthetic strategy, if you can see a way in which you can put a symmetrical prochiral molecule into your synthesis, that can help you with your overall yield. So what we've done so far is talk about resolutions. What I'm going to talk about now is how we exploit another aspect of enzymes, which is chemoselectivity. So the molecule I'm showing you is a hypothetical molecule. This isn't an actual experimental example, but I've constructed this so that because it really exemplifies the kind of selectivity that you can get with enzymes. We have on this molecule an ester group, a cyano group, and an Na-sal group. So if you want to selectively cleave one of these groups, for example, the ester group, we can select an ester hydrolase, and that will selectively take off the ester group, the O-acetyl group, without touching the N-acetyl group or the cyano group. Okay, maybe that's not that clever. You could probably find conditions with chemical catalysis which would achieve the same ends, just by careful control of reaction conditions. But what if you want to take off the Na-sal group whilst leaving the OA-sal group in position? That's probably a bigger challenge. But if you use an enzyme, you can use an Na-salase or a protease. It's quite easy to achieve that, and you'll get very clean deprotection of the nitrogen, leaving the O oxygen group protected. Perhaps even more interesting is when you look at the nitrile group. Using a nitrile hydratase, you can hydrolyze the nitrile through to an amide without touching either the O-acetyl or the N-acetyl. And if you want to take the nitrile all the way through to the acid, you can use a second enzyme, an amide hydrolase, which again will be very specific for hydrolysis of the amide to the acid. In fact, what you can do is use a nitrolase, which will take you directly from the nitrile to the acid, if that's what you want to achieve. So what I'm trying to flag up here is that enzyme catalysis facilitates precise modification of polyfunctional molecules. It can give you more direct routes to your target molecules with less requirement for protection and deprotection. It simplifies your overall synthetic route. And these kind of things, these concepts need to be built into the original routes selection exercise. So all the reactions I've talked about so far have been related to ester hydrolysis or transesterification, that kind of reaction. What I want to talk about now is redox reactions. So when we do redox reactions in biological systems, and I'm going to refer to a ketoreductase reaction here to exemplify it. What we're talking about here is taking a ketone through to a hydroxy compound. That's catalyzed by the enzyme, which picks up its reducing equivalents from one of these compounds called nicotinamide adenine dinucleotide cofactors. So NADH2 is a complex cofactor which donates a hydride to the ketone and delivers the hydroxyl as a product. In doing so, it's oxidized to NAD, its oxidized form. And these cofactors are essential for use in a biological reaction. The only problem with them is that they're very expensive. So NADH costs about $1,200 a mole. NADPH is even more expensive at about $5,500 a mole. So you can't use these things stoichiometrically. So the question is, how do we get around this? Well, one very simple way to get around this is to use what we call a live cell system to recycle the cofactor. So the little cartoon that I'm showing here, the box is meant to represent a live cell. Inside that cell, all the enzymes necessary for supporting life. And the organism is taking up glucose, metabolizing it to carbon dioxide or water, and in catalyzing those oxidations, the reducing equivalents are transferred onto the nicotinamide cofactors. If you then supply a ketone substrate to that culture, then if there's an enzyme inside the cell which can accept the ketone as a substrate, it should be reduced to give you your target compound. And this is a nice and simple way to screen for a particular activity that you might want. So the advantages of this live cell system is that you don't have to add these expensive cofactors exogenously. The organism makes them themselves from simply something like glucose, which is nice and cheap. And it's a nice simple way to screen for the enzymes that you're looking for. But as with all things, there's downsides as well as advantages. And on the disadvantages side, we've got a requirement for fermentation, not always available to the process chemist. It's a black box system as well. We don't actually know which enzymes catalyze in the reaction of interest. And the other thing is that sometimes the substrate or the product can be toxic to the cell. And in order for this system to work, the organism has to be viable. It has to be living. So toxicity is something we have to be aware of and take care of. Despite these limitations, these kind of systems have been used on a number of occasions and still are used for manufacturing scale keto reduction reactions. What I'm going to do now is give you a specific example that we've developed and talk you through that. So the actual example refers to the synthesis of an intermediate called hydroxysulfone, which is used in the synthesis of a drug called Dorzolamide, which is an anti-glaucoma drug. Hydroxysulfone itself is derived from the precursor ketone, the ketosulfone, and I'll point out the fact that there is already a chiral molecule, chiral center in that ketosulfone, the methyl group. That is also derived by biocatalytic reaction. I'm not going to go into the details of that today because we don't have time, but it is relevant to the subsequent chemistry. So what we're trying to do is to reduce the ketosulfone to the hydroxysulfone. You could argue that we can do this chemically, and in fact you can. The problem is that the existing chiral methyl group results in diastereoinduction. And it actually is one of those occasions where it works against you and you end up getting a very high selectivity, but for the wrong diastereoisomer. So we end up with hydroxysulfone with the wrong, very poor diastereomeric excess. The question is, can we find an enzyme to do the reaction with the opposite diastereoselectivity? So what we did was to use a live cell catalyzed system to screen for this particular activity. And we were able to identify a number of organisms which would do the reaction with very high selectivity. The one we selected was a fungus called Neurospora crassa. It was a fermentation-based bioreduction system where we grow the organism, feed in the ketosulfone, feed in glucose as a carbon source to drive the reaction. And we get a very clean conversion, very high selectivity, around about 99 to 1. This process has been routinely operated at plant scale. And at the end of the reaction is simply an organic extraction of the, organic solvent extraction, crystallization, and we can recover the hydroxysulfone in around about 85% yield. And it works very well. It's been operated for many years, and you could argue, why would we want to change it? Well, one of the problems with the whole cell fermentation type approach is that you do need access to fermentation capacity, and that's not always at the same site as the rest of the chemistry. Much better if you can uncouple the fermentation aspect of the process from the chemistry side. And so what you can do to achieve that is to take the enzyme from the organism and then use the enzyme as an isolated protein rather than the whole cell. And that's what we've done with this process, and we've converted it into an isolated enzyme reduction. But what we end up with is the ketosulfone reductase. We've cloned the gene from Neurospora crassa, overexpressed it in E. coli, so we can make lots and lots of enzyme in a small fermentation volume. We can then store that, and then we can use it whenever we want it. So we've decoupled the reaction of the ketosulfone to hydroxysulfone from the fermentation stage. So that helps with logistics. The other thing is that because we're not working with a live system anymore, we can do these reactions in standard reactors. It's much more like a standard chemical reaction now. And in this case, the cofactor recycle system is put in place by the use of a glucose dehydrogenase enzyme, which oxidizes glucose to gluconic acid and thereby reduces the oxidized form of the cofactor NADP back to the reduced form NADPH. So ultimately, oxidation of glucose drives the reduction of the ketosulfone. So we're using an inexpensive hydride donor effectively in the form of glucose. The process is operated at 100 kilogram batch scale. Much more like a chemistry process now. The actual reaction doesn't involve any microbiology per se. That's all done separately by microbiological experts, fermentation experts. And it fits in much better with the chemical synthesis environment of the process chemist. So the next example I want to talk to you about is an example where we've put together a novel synthetic pathway involving two biological steps to overcome some particular synthetic difficulties that we're facing. The compound in question is a statin intermediate. It's this compound here, BHA. It's an intermediate in the synthesis of AstraZeneca's rosuvastatin. It's a relatively simple molecule, BHA, but it does present some interesting synthetic challenges. So what I'm going to do next in the next slide is to run through what we came up with. The first thing to say is that there's been an awful lot of interest in the statin intermediates. There's an absolute minefield of intellectual property out there. So not only did we have to come up with a synthetic route which avoided existing intellectual property, it also had to be cheaper than anything that was already in place. So it was quite a challenge. So let me talk you through that step by step. Our starting point was this molecule shown here on the top left. The existing chiral center there was easily accessed from hydroxybutyrolactone, which was essentially a chiral pool material derived from sugars. It was available in bulk very cheaply. And by reacting that in a Claisen reaction with tert-butyl acetate, we could get this C6 ketoester. So the next step was to reduce the keto group to give us the chiral diol that we were looking for. And initially we looked at using asymmetric chemical catalysts to achieve this, and we had some success. But the best we could achieve was about a 9-to-1 ratio. So quite respectable, but the problem is that all these intermediates, they're high boiling point oils, not easily crystallizable, and therefore separating the two diastereoisomers at commercial scale was going to be difficult. We couldn't use chromatography, it would have to be something like a crystallization. So could we find a more selective route to this reduction? And the answer is yes. We managed to identify a yeast, Pichia angusta, which would reduce this to essentially a single diastereoisomer. And the scaled-up process would do this reaction with 92% yield. So very, very selective. And from here, in principle, all we have to do is to add the acetonide group, and we're there, we've got BHA. But as in all things in life, things turned out not to be quite that simple. When you try and put the acetonide on there, not only do you get the 3,5 acetonide, which is the one you want, but you also get reactivity with the primary hydroxyl at the 6th position to give you some of the 5,6 acetonide. And again, as I mentioned, these compounds are very difficult to separate, not easy to crystallize, so we had a real problem. So what are we going to do? Well, this is where we fell back on the regioselectivity of enzymes, and we set about looking for a regioselective protection of that primary hydroxyl group. And again, our old friend Candida antarctica lipase B came to the rescue. We were able to catalyze a regioselective esterification of that primary alcohol in toluene using vinyl acetate as the acyl donor. Very good reaction, very clean, very selective. 94% isolated yield here. In situ, in fact, we don't actually isolate this intermediate. So sorry about that. So 94% in situ yield, which can then carry through to the acetonide formation reaction, totally selective, giving us the 5,6 regioselective, the 3,5 selectivity that we're looking for. And the bonus that we get from this route is that this compound is easily crystallizable. So we can purify at this stage, clean it up, and then do a simple hydrolysis to give us our target compound, BHA, in overall about 60% yield from our starting keto ester material. So this has been scaled up, operated at multi-ton scale, and it's now a patented route. So the next example I want to introduce you to is, again, a very similar redox reaction to the ones we've already seen. Again, we're operating in an isolated enzyme format, but this time we're making an amino acid, an alpha amino acid, from the corresponding alpha keto acid. So the enzymes, amino acid dehydrogenases, present in biological systems, will catalyze this reaction. And by setting up cofactor recycle in a similar way to the previous example we talked about, we end up with the scheme shown here, where the oxidation of formate drives the reduction of the oxidized form of the cofactor, and the amino acid dehydrogenase then catalyzes the reductive amination of the keto acid to give you the amino acid. And this is a versatile and efficient route to a range of unnatural amino acids, where the R group shown here can be anything you can think of, pretty much. It says in the slide S amino acids, but there are also amino acid dehydrogenases available now, which will also react to give you the R configuration. So it's a very flexible and very versatile approach to amino acids. So what I've talked about so far is some mainstream biocatalysis using pretty standard types of enzymes. What I want to do for the rest of the presentation is to introduce you to some more esoteric enzymes, which are probably less well known to some people, just to give you a taste for the breadth of the type of things that enzymes can do. So the first one I want to talk about is an enzyme called dimethyl sulfoxide reductase, and the application of that to the resolution of racemic sulfoxides. So DMSO reductase is an enzyme that enables certain bacteria to live in the absence of oxygen. Most what we call aerobic organisms, things which rely on oxygen to live, use oxygen as the final acceptor for electrons from the oxidation processes that are used to liberate energy. If oxygen is absent, you have to put those electrons somewhere else, and certain organisms have evolved a system which enables them to put those electrons onto dimethyl sulfoxide, liberating dimethyl sulfide as the product. So the question that we asked ourselves is, can DMSO reductases accept other sulfoxides? And if it can, and these sulfoxides happen to be racemic, will they demonstrate enantioselectivity? So a range of DMSO reductase producing bacteria were obtained, things like Escherichia coli, Rhodobacter capsulatus. These are already known to possess DMSO reductase. But what we also did, besides obtaining those from culture collections, was to try and isolate from nature some other organisms which also produce this enzyme. And we did this by using a process called selective enrichment. So basically we took soil samples, put them into a nutrient medium, incubated the culture under anaerobic conditions with various sulfoxides provided as potential electron acceptors. Anything that grows is probably using the sulfoxide as a terminal electron acceptor. And you can tell by the smell of DMS, for example, that DMSO is being reduced. So it was very easy to select for organisms with these activities. So having obtained a range of organisms which for putative DMSO reductase producers, we decided to screen them against a model compound. And we chose this structure here, methyl paratoluol sulfoxide, because it was commercially available. It was available in both enantiomeric forms, so it was a good model for our tests. What we were able to show is that Rhodobacter capsulatus and one of the serraceous species which we isolated from soil was able to resolve this compound with very high selectivity, giving us about 98% EE at just over 50% conversion. In contrast, two other organisms, Proteus vulgaris and Citrobacter brachii, we were also able to do the reduction of methyl paratoluol sulfoxide, but in this case, they reduced the other enantiomer and left behind the S enantiomer. So by selection of the appropriate biocatalyst, we could get access to either enantiomer. Now, methyl paratoluol sulfoxide is not a particularly interesting molecule to us, but we did have some other compounds that we were looking at which were of commercial interest. So we decided to screen some of our organisms against these structures to see what happened. So initially, we looked at this Citrobacter brachii, and we were able to demonstrate that the ethyl pyridyl sulfoxide shown here was resolved with high selectivity. And this thiosulfonate structure was also resolved with selectivity. And in fact, the brachii enzyme seemed to be the only one of the ones we isolated which would react with this substrate. But perhaps the most interesting structure was this one here. This is omeprazole, a proton pump inhibitor marketed by AstraZeneca, initially marketed as a racemate, but subsequently switched to a single enantiomeric form. And we were able to demonstrate that a number of these organisms expressing sulfoxide reductases were able to very selectively reduce one enantiomer of omeprazole and also of some of the other analogous proton pump inhibitors. So I think the reason I'm showing this example is because I think reaction of sulfoxides is not a very common one in synthesis, but it just illustrates the breadth and versatility of what can be done with enzymes. So the next example that I want to introduce you to is enantioselective dehalogenation. Now, halogens are not found very commonly in biological systems, but enzymes do exist which are able to dehalogenate organic halo compounds. And the reaction of interest to us was the dehalogenation of 2-chloropropionic acid. What we were targeting was an enzyme which would take the racemic 2-chloropropionic acid shown on the left here and hopefully selectively hydrolyze the R enantiomer to lactic acid and leave the S enantiomer unreacted, the S enantiomer being the one that we wanted to use. So S2-chloropropionic acid is our target. And because there were no commercially available dehalogenases that we could go and purchase, we had to look for them in nature. So again, we used this selective enrichment procedure where we provided 2-chloropropionic acid as the sole source of carbon and then tried to get organisms which would grow on that. And since the reaction concept was that we were looking for a hydrolytic dehalogenation, the product of the reaction would be lactic acid, which is a common growth substrate for bacteria. If we could find an organism which grew on 2-chloropropionic acid, it was probably in possession of a halo acid dehalogenase, producing lactic acid and thereby growing. So by using this approach, we were able to isolate a number of organisms which produce these enzymes. And from this initial library of dehalogenases, we were able to identify some which were specific for the R halo acid and some which were specific for the S halo acid, and in fact some which were unspecific and would accept either. Obviously for our process, we were looking for the R-selective enzyme, and we cloned the enzyme, or the gene for that enzyme, overexpressed it in E. coli, and developed a manufacturing process from it. So we now use this halo acid dehalogenase to synthesize over 1,000 tons per annum of S2-chloropropionic acid. So it's a very powerful technique to use selective enrichment if you can set up the right conditions. So we have not only the R-selective enzyme, which provides the S enantiomer, but we also have S-selective dehalogenases, which will provide the R enantiomer. So having got these enzymes, we were interested to know whether they could do anything else. Can we use them to make any other interesting compounds? And the answer is yes. So the enzymes will work with 2-halobutyrates to give us, for example, 2-bromobutyrate or 2-hydroxybutyric acid. We have enzymes to do both enantiomeric series. And they also work with 2,3-dihalopropionates. to give residual substrate of 2,3-dibromopropionate in optically pure form, and the hydrolyzed product, which is a 2-hydroxy-3-bromopropionate. And what's particularly interesting about this one is that the product of the hydrolysis has 3 different functionalities on 3 contiguous centers. So you have a carboxyl group, a hydroxyl group, and a bromine, all adjacent to each other, giving you the possibility for selective derivatization. So quite a versatile small synthon. One of the disadvantages of these dehalogenases is that the substrate range that they accept is quite narrow. But enzyme evolution offers a potential for expanding that substrate range. Directed evolution of enzymes is well established and could be applied to these enzymes to expand the scope of dehalogenation. So that really is a quick run through of some things that we can do with biocatalysis, starting from things we were doing back in 1990 through to more recent examples. But the question is, where is biocatalysis today? And I think the answer is that biocatalysis is now considered during the initial route selection. You know, synthetic route is often built around a key enzymatic step. So, you know, the organic chemists will get together with the biologists and they'll put their heads together and try and construct a route bearing in mind where an enzyme can be used. This is something that simply didn't happen 20 years ago. A large number of drugs are now synthesized using biocatalysis. There are examples in the statins market, anti-diabetics, anti-HIV, and we're talking about multi-ton manufactures here, tens or even hundreds of tons of manufacture. So biocatalysis is becoming the first choice compared to conventional routes such as diastereomeric crystallizations and even asymmetric chemical catalysis. And this has all been driven by advances in the biological sciences which have underpinned this growth in biocatalysis. The biological sciences have gone through a revolution in the last 25 years, and this has really pushed forward what we can do in terms of applying biological catalysts in chemical synthesis. So what kind of advances am I talking about here? In terms of biocatalysis, the critical advances is quite simple. It's access to a much broader range of enzymes at commercial scale. At the beginning of the talk, I was talking about access to maybe a library of 10 enzymes to find something we're looking for. Today, you're talking about hundreds of enzymes. Different reaction types are available, so ketoreductases, ene reductases, monooxygenases, talked about sulfoxide reductases, dehalogenases, the list goes on. So the list is as long as your imagination, really. Within each of those classes, there's multiple variants of enzymes, some which will give you the S enantiomer, some which will give you the R enantiomer, some which will work with different substrates. So you can spread your search until you find the enzyme with the reaction, with the selectivity that you're looking for. So much increased range of substrates and enantioselectivities are available. So how have these advances occurred? Well, it's a result of advances in genetic engineering primarily. Rapid genome sequencing, publication of genome sequences. These days you can sit at your computer, look at a database, pull out a putative sequence, and you can then send that sequence to a company that will actually synthesize it for you. Gene synthesis technology has advanced massively, and it's now very cheap. So you can send a gene sequence to one of these service companies. They can synthesize that gene for you. They can send it back to you within a couple of weeks, and the whole process costs you about $500. So this is now beginning to replace the old-fashioned conventional gene cloning methodology for many applications. And our understanding of the relationship between the structure and function of enzymes is expanding massively as well. Many more crystal structures are coming available. We can understand better what changes need to be made at an enzyme active site if we want to change its selectivity. And we can then apply evolution techniques to modify those enzymes. So having said all that, what is the key to success in biocatalysis? Well, I'll give you my opinion. The first thing is you need to have a broad and deep knowledge of biology. The underpinning science here is all biological. We need to understand metabolism and biochemistry. We need to understand genetic engineering, enzymology, and fermentation. But ultimately, what we're doing is applying these biological systems in the sphere of organic chemistry. So we need to be able to think like an organic chemist during the root invention. Ultimately, we want to scale it up. So we need to be able to think like a process engineer during the process development. And clearly, none of us are expert in all of these areas, but we do need to have enough of an understanding of the organic chemistry and the process engineering to be able to talk to each other, understand each other's problems. And that means we need to be able to work as a multidisciplinary team. And it's stringing all those things together that enables you to put together the overall package of being able to take a biological idea, put it into a chemical environment, and then ultimately deliver a practical process that can be put on a plant by the process engineers. So that's the end of my presentation. I hope it's given you some feel for what we can do with biocatalysis, and I'm happy to take any questions in the short time that we've got left. So I'll hand you back to Rachel.
Okay, thank you very much, Bob. So thank you to everyone who has already sent their questions in. Please continue to send in your questions via the Q&A widget at the bottom of your screen, or please use the top left-hand box. We will try and get through as many of the questions as possible. However, if we don't have time to answer them all in this webinar, then Bob will get back to you at a later date. So, for your first question, as a contract developer and manufacturer, how do you deal with any intellectual property that emerges during work on a customer molecule?
Okay, right. I think the first thing to say is that the customer has to have comfort in where they stand on this. So if a customer is funding some work with us, then any arising intellectual property would belong to the customer. If it's work that we're doing ourselves, funded out of our own pocket, then obviously we would want to have access and control over that. But clearly the client needs to have some comfort that they have control as well. So we wouldn't want to use it as handcuffs over any client. We like to work in a collaborative manner with our clients, and the only way to be successful in this game is to have a win-win situation. So really, it has to be an open discussion between ourselves and our clients, such that both are comfortable and everybody gets what they need. If you can't get the win-win situation, then what you end up with is a lose-lose situation. There's no such thing as a win-lose in this. So everybody has to get what they need, and that's what we're targeting.
Okay, thank you very much. And we read a lot about enzyme immobilization in the literature. What is your opinion on its value?
Okay, immobilization has been talked about for an awful long time, going back to the 1960s, 1970s. There's not a simple one to answer this. There are a number of areas where you might want to use immobilization. The first is if the enzyme is difficult to access or very expensive, immobilization gives you the opportunity to recover and reuse the enzyme. And that is used particularly in very large-scale processes like penicillin synthesis, where the penicillin acelases are immobilized and reused many, many times. For a very large-scale process like that with a very stable enzyme, that makes good sense. Another reason would be if you're carrying out a reaction in a water-immiscible organic solvent, such as a transesterification. Most enzymes do not disperse well in organic solvents, but if you immobilize it as a monomolecular layer on an inert particle, as a carrier, then you can expose much more of your enzyme active site to the reacting medium. You get better dispersion, and this gives you orders of magnitude higher reaction rates than you would otherwise get. A third reason for doing it is if you have a purification issue in terms of having to get protein out of your reaction. If you've got it immobilized on a macroscopic particle, you can simply filter off your biocatalyst. I'm not aware of many cases where that's been an issue, but it would be a situation where immobilization could be successful. But at the end of the day, it's always a cost-benefit consideration, because the cost of immobilizing has to be taken into account. Enzymes are pretty cheap to make these days, and very often it's simpler and cheaper just to use it as a protein solution and discard it at the end of the reaction.
Okay, thank you very much. And how does biocatalysis compare with conventional chemistry in terms of the time taken to scale a reaction to plant scale?
Okay. Generally speaking, I would say they're comparable. It depends on what you take as your starting point. If you have an enzyme available, so let's say we're using a commercially available lipase to catalyze an ester hydrolysis reaction, for example, then scaling it up is very straightforward. And in terms of comparing it to a diastereomeric crystallization as a competing route, I would say that the enzyme route would probably be quicker to optimize and scale up. So I think we shouldn't be concerned that scaling up a biological-based reaction is necessarily going to be more time-consuming than a chemical route. If you need to discover a novel biocatalyst to catalyze your reaction, then clearly the time frame will be longer. But then if you have to develop a novel chemical catalyst for your reaction, again, you're building in extra time. So I would say, generally speaking, I wouldn't be concerned that biological catalysis brings in extra time in terms of the lead time to scale up.
Okay, thank you, Bob. So actually, unfortunately, that is all we have time for in today's session. So to the attendees, you will receive an email shortly telling you how you can access the on-demand version of this webinar, or you can access this through our website, which is www.business-review-webinars.com. I know we did have a few questions come in about accessing that. So yeah, as I said, sadly, this is all we have time for in today's session. So if we didn't get round to answering your question, then Bob will get back to you at a later date. So that just leaves me to thank Bob for what was a great presentation, and thanks to Piramal Healthcare for sponsoring this session. We look forward to sharing further webinars with you, so please do keep an eye out on the website just mentioned, and follow us on Twitter at BRWebinars for daily updates, and join our LinkedIn group, Business Review Webinars. So thank you once again to everyone, and I hope you all have a nice day.